My name is Brett Branco. I'm the executive director of the Science and Resilience Institute at Jamaica Bay, and this is our Jamaica Bay Colloquium session on fish populations. So the institute, the SRIJB, is a CUNY-wide institute hosted at Brooklyn College, and we work with New York City, the National Park Service, and an extensive network of partners to support the well-being of people and nature in and around Jamaica Bay through research, education, engagement, and training. So today's colloquium is part of a series of events on the state of Jamaica Bay that are being held in partnership with the Jamaica Bay Rockaway Parks Conservancy and the Jamaica Bay chapter of the American Littoral Society. The purpose of these colloquia is to explore current research and data that informs us of conditions and their trends in and around the Bay. So now I'm going to turn things over to Alexandra Kononik from the American Littoral Society, who is our one of our co-host and partner, and she is going to introduce the speaker and facilitate the rest of this webinar. So over to you, Alex.
Thank you so much, Brett. I'm so excited about today's talk. Let me introduce you all to our speaker for the day. Dr. John Waldman is professor of biology at Queens College, and he is a heavy hitter when it comes to his knowledge, expertise, and contributions to the science and our understanding of diadromous fishes in New York Harbor and the greater Hudson River estuary. John has written many articles, 110 scientific articles, technical volumes, and has occasionally been a contributor to the New York Times. John has authored many books and publications on the state of New York City's waters and aquatic environs. Two of his books, Heartbeats in the Muck and the subsequent Running Silver, Restoring Atlantic Rivers and Their Great Fish Migrations, I would say are core reading for anyone who is an interest in fish and wants to understand the current and historical drivers behind the changes and challenges fish populations have faced in New York Harbor and our rivers in the Northeast Atlantic region as a whole. Today, Dr. Waldman will be speaking on his observations in Jamaica Bay on climate and fish communities in the surrounding region. And with that, John, we hand it off to you.
Thank you, Alex, for that generous introduction. So I'm going to speak today about climate stress and the regional fish fauna, something I've been very interested in. I've been watching our local waters for a very long time, many decades, and I think there are some very interesting things going on, visible in Jamaica Bay and also elsewhere around the harbor. So next slide, please. So we have a very highly diverse ichthyofauna. Briggs and Waldman in 2002 came up with 338 fish species, not including the Hudson River. And they are in various categories like pelagics and migratory coastal, resident, diadromous, and so on. And they are, for a temperate water body, a really quite diverse set of fish in terms of the evolutionary history and their habits. There's also about 220 that have been taken in the Hudson River itself that Tom Lake, who edits the Hudson River Almanac, has documented through time. Next slide. So one way we think about fish distributions in anything that's coastal is in terms of zoogeographic provinces. These are regions that share commonalities that tend to bring similar species together within that province that are somewhat distinct from surrounding provinces. They're not perfect barriers. The barrier at Cape Hatteras, the barrier at Cape Cod are certainly passable, but they are kind of like rough fence lines that help guide these fish populations. So the Jamaica Bay is squarely in the Virginia province, and a lot of these fish have been seen and recorded in the Fishes of the Gulf of Maine, which is a misleading title because even though it focuses on the Gulf of Maine, it's a very rich source for all kinds of observations on these species. Next slide. So water temperatures have been rising. There's, you can argue about some biological responses, but a thermometer in the water is very clear cut. It is what it is. And there's been almost a 1 degree Celsius increase in the Hudson River at Poughkeepsie, which is a very well monitored site between 1946 and 2009. And that's not a small change. Next slide, please. Another long-term data set, although not nearly as long-term, is the New York Bight Indicator Report from SUNY Stony Brook and it shows a steady increase in summer temperatures in this region. Next slide, please. So there are some possible metrics of change in relation to climate, and they're not easily separated from each other. They're correlated in some ways. One is just abundance. Another is distribution, both horizontal and vertical. And the third is phenologies. And these signals are often confounded, and I'll try to separate them a little. Next slide, please. So here's a winner in terms of warming. This is striped bass in the Gulf of St. Lawrence. Striped bass in the Gulf of St. Lawrence were always a very kind of minor population in the lower part of the Miramichi River. And over time, they started to explode in numbers. I would say about 10 years ago, they really became very, very numerous. And all of a sudden, which is typical of a species that has an expanding population, they expanded their range. And you see in the lower left, the source of the fish, the Miramichi River. And you can see in the upper right, that red circle, that's as far as they went in their peak year of 2017. The population exploded from roughly 5,000 individuals to about a million. And when you have that kind of change, you have density dependent processes that push fish further away and spread out. So there's a clear climate signal not that far north of us. Both the Gulf of St. Lawrence and the Gulf of Maine are experiencing some of the fastest water temperature rises in the world. But so we had a winner. The striped bass exploded in numbers. At the same time, the Atlantic salmon, which is revered by many anglers in that part of the world, started to go down, partly because of predation by striped bass. And in fact, as much as we revere striped bass in our local waters, you can see in the lower left that little image showing that striped bass are, public enemy number one because they eat salmon. So these are the kinds of societal conflicts that originate from vested interests being challenged with new biologies in effect. Next slide, please. So phenologies are hard to actually quantify. A good job was done a while ago in the Penobscot River for salmon. Between 86 and 2001, they found that salmon were entering the river about 1.3 days earlier per year. In the Androscoggin River in Maine for alewives, similar time period, about 1.2 days earlier per year. So these timing events are significant because timing events are related to all kinds of factors as these fish move in. It could be, competition and predation, the time the eggs are laid in terms of when they will hatch and be able to enjoy the plankton to then mature into a cohort, so on. So phenologies are shifting, and they are not the easiest thing to study because a fish run may occur in sporadic spurts over a period of time, and it's hard to get a grip on the actual movements over time. Next slide, please. So we could think about climate in our waters in terms of possible refuges and overall effects. Rivers provide one refuge, which is to go further upriver where temperatures are typically cooler in rivers. But there's a finite distance that will work. You can't send huge fish up tiny rivers. So rivers are a refuge for certain size classes of certain species. Lakes are a refuge for species that live in lakes if they have two-story stratification. If they're a shallow lake and the waters become very warm, there's nowhere to go. But some lakes have summer stratification where the bottom waters are much colder, and that is a refuge. Anadromous fish have extirpations and colonizations along the coastline. I'll show you what I mean in a minute. And marine fish can go north or go deep. So in terms of the freedom of action and response to climate change, marine fish have the greatest flexibility. Next slide, please. So this is an interesting comparison. These are the rivers of the United States, and we see a suite of fish in the Gulf of Mexico and a suite of fish on the East Coast that look very similar. And in fact, they are very similar, although they're taxonomically interesting in the diversity. The striped bass is the same species on both coasts. The Atlantic sturgeon is a subspecies on both coasts, and then the shad are two separate but closely related species. But the point is that the Gulf of Mexico fish have no refuge. They are up against rivers that are coming in from the central part of the country, and they have nowhere to go, east and west to cooler water. Whereas the East Coast fish, their counterparts, are able to retreat northward and give up rivers to the south and gain rivers to the north to a certain degree. Next slide, please. So here's some examples of anadromous fish facing climate. Rainbow smelt are kind of a silvery species that lived in the Hudson River until about 1998. Before that, people would dip net them in their backyards and catch them in huge numbers. They're very good to eat. And their numbers were tracked with the Hudson River Utilities Survey. And you see those blue bars on the left. Those are annual catches of post-yolk sac larvae over that period of time. But then around 1998, they just crashed. I know they show it a little earlier here, but I think the last specimens of this species, which is a true cold water species, this is a fish that likes to spawn in small tributaries of the Hudson in February and March when waters are still cold. They disappeared, and the last one taken was in 1998. So there's a loss of a species that I think is directly attributable to climate change. Next slide, please. Another species that is similar in its temperature requirements to the smelt are Atlantic tomcod. And tomcod have been monitored in the Hudson River Utilities Survey for a long time. And between 1974 and 2016, they found that the peak age ones were in 1976 at about 8.5 million, and that the lowest number was only about 5,000 in 2012. The last time they exceeded a million was in 2004. And you can see in the graph on the lower right that they start to get a little more wobbly in their numbers. They're shown in blue, and actually yellow and blue, and they vary more as time goes on. And this is a signal often when a population is under stress. The annual abundances will change as the system sort of breaks down. So we still have tom cod, but they become very scarce. And I do expect that one of these years they'll join smelt on our list of long gone species. Next slide, please. True marine fishes have the most freedom to adjust to temperature. They're not so rigidly geographically fixed. They may spawn on a particular reef, but they can give that reef up and move farther north if need be. On the left, it shows an image of how cold water and warm water species are behaving in relation to temperature change, temperature increase in Narragansett Bay. And you can see that over time, from year to year to year, that the cold water species are being ratcheted down to a shorter season of inhabitation of these waters, and the warm water species are gaining more time because of the warmth of these warming waters. Another really good survey, which I don't have any plots for here, is by Janet Nye et al., who looked at NOAA trawl surveys from 68 to 2007 along the North Atlantic coast, and she found that 24 or 30 fish stocks showed movement towards the poles or deeper water or both. And these are the refuges for true marine fish. You can move north, you can go deeper, or you can use a combination to survive, up to some point. Next slide, please. So we can look at these fish also in qualitative ways. I've been an angler and a close watcher of our fisheries since I was, 6 years old, and a lot older than that now, many decades behind me. And I've seen a lot of changes that, occurred in my lifespan. One of them, in the upper left, we have the silver hake. This was a fish that was routinely caught on the South Shore of Long Island in the winter. Sometimes they would chase bait fish right up into the surf, and people could gather them with their hands on cold winter nights. They are largely gone from this region. Below that, you have the window pane flounder, which is a very, very thin flatfish. It's called window pane for a reason. You can kind of see through it. People used to call them sundials, and They are a winter species that we hardly ever see anymore. On the lower right, we have mackerel. And mackerel, I thought they were gone about 20 years ago. They had not really made a run in our region for a very long time. And then I think it was 2 years ago, we started seeing some young of the year again. So one of the observations about these changes is that they're not automatically unidirectional. You may get some shifts for unknown reasons. So mackerel came back for a year or two, and now I think they're mostly gone again. And in the upper right is a really beloved species, the winter flounder, which is, great tasting fish that was sought by anglers in the spring and in a fall season. And their numbers started to decline. And over time, the state, New York State, ratcheted down the open season. They got rid of the autumn season and they shortened the spring season, and they put in length limits, and the numbers just kept getting lower and lower and lower. And my friend and colleague, Ike Wergen, who's a fish person, told me once he went to Cold Spring Harbor on Long Island and he and his buddy caught over 200 flounder in one day. I would say that if you fished every day for a few weeks in flounder season now, you might be lucky to put a catch of 20 together. They're just really, really scarce. And they're cold water species that's just retreating north. Next, please. One distinct, but apparently, kind of related phenomenon is the fact that we get tropical or semi-tropical fish in warm core rings along the Long Island coast. So you see there are these warm core rings out off the ocean shore, quite a ways out, and they are carrying warmer water from the south. And that warmer water is carrying eggs and larvae of tropical and semi-tropical fish. And these warm core rings will, in some cases, actually sort of strike Long Island and deliver all kinds of tropical fish that you would not expect to see up north. And that includes things like you see in the upper photo, look downs, or below permit, and to the right, grouper. And there's actually a very large number of these that come up every year, but they only come up as young of the year. They either die up here when the waters get too cold if they can't migrate back south, or they retreat to the south and don't come back again. So this is an ongoing phenomenon, and it's remarkable, especially on the South Shore, which is the area that receives these fish, because the North Shore is bounded by Long Island, and these warm core rings don't get there. The South Shore, there can be amazing numbers of these tropical fish. I went out with Todd Gardner once, who is the premier tropical fish collector in Long Island, and we spent a day doing everything from snorkeling to seining to angling, and we had about 30 species of fish, and half of them were tropical fish. So this is an interesting phenomenon, but it's not, from what we can see, a product of climate change. This has been going on a long time. And it's just interesting that nature delivers these fish that to the north that I think in many cases don't survive. Divers have seen these fish dying on the bottom of the waters when it gets cold. Next slide, please. So one of the serious consequences that's worth mentioning in terms of temperature and climate is lowered oxygen and slower fish growth. It's a fact that as waters get warmer, they cannot hold as much oxygen, and a 1 degree Celsius decline can cause a 30%, this is covered up a little, it should be 30% decrease in growth for fast moving fish like tuna, and also for 18% for slow growing fish like brown trout. So as our waters are warming now, we're going to see and eventually be able to detect smaller individuals in many cases for mature fish. Next, please. And this is an example from my friend and colleague, Karin Lindberg, looking at oxygen in relation to temperature, which is an inverse relationship. And you can see from this plot that as temperatures are rising, dissolved oxygen is going down. And that's going to have consequences for growth, because individual fish that are in these lower oxygenated waters have to work harder to pull the oxygen out of the water, which diverts energy that would have gone to their somatic growth into simply respiring. So this is a serious issue. Next slide, please. And I've been fascinated by this photograph from the Delaware River from the 1800s, because it shows one day's catch of shad, American shad. And on the right, you see some images from current shad festivals. And I looked at some of the data for these shad festivals. A typical winter, the biggest shad for the whole festival will be about 4 pounds, 11 ounces, 5 pounds, 2 ounces. And I would say the vast majority of the fish that are being hung here in the photograph on the left are bigger than that. So there's been a dramatic decline in shad size. And I don't know if you ever want to read a great book about shad, John McPhee's book, The Founding Fish, is worth picking up. And he said that in the year 1800, shad packed about 40 individuals to the barrel, and the year 1900, about 100 individuals to the same size barrel. So we're witnessing this major decline in growth of these fish, and it's not entirely clear why or what its ramifications are. Next slide. So one of the ways that we can look at sort of constellations of fish species are, um, and also their habitats are with regime shift analyses. And the regime shift analysis that I like best of all those I've encountered is the one from the Baltic Sea, because it really shows a very clear pattern. This is not numbers of fish. This is on conditions that these fish live in. And you can see that there are 75 time series variables, which is an enormous number. And that in the upper left of that figure, that circle on the left is showing the 1970s, a time when there was low temperatures and high salinities. And then you can see it jumps in 1988 to another circle, which is characterized by freshening of the waters, warming, and anoxia on the bottom. And then thirdly, there's on the upper right, a pattern where you have you have warm waters, low salinity, and stable nutrients. And what's really interesting about this is that these particular regimes are so discrete from each other. They are not mingling. You have completely different patterns that stand out in these data. So my colleague Jose Anadon and a couple other people, next slide, please, did a study of the Jamaica Bay DEC SANE survey, which is a fabulous source of data. They had 29 years of fish surveys that were conducted using typically 600-foot long beach seines. So they scooped up a lot of fish, and in 29 years, they captured 834,000 individuals of 76 species. So this was a fabulous database. And along with some colleagues from DEC, like Jesse Hornstein and a couple other people, we dived into this using multidimensional scaling and also looking at the patterns that would emerge in two ways. One was with transforming the data using something called the Hellinger transformation, which downweights the effects of rare species, and then just using the raw non-transformed data, which is more sensitive to rare species. So next slide, please. So we published this in Ecosphere, and I have to say it's a really, really complex and interesting paper, mainly because Jose Anadon is sort of brilliant at this kind of analysis. And he titled it Decoupled Shifts of Dominant and Rarer Fish Species as a Response to Warming and Extreme Events in a Large Estuary. And next slide, please. And there's lots of output from this. And if you have an interest in the subject, I urge you to find the paper, write to me, I'll send it to you. But just to give you a taste, with the Hellinger transform data, we found that menhaden, and all these fish that I mentioned here with the plus or minus signs are ones where you had linear trends or at least unidirectional trends. So with a Hellinger transformation, which downweights rare species, we found that menhaden had a 6 order of magnitude increase in abundance over that period. Toadfish, a 2 order of magnitude increase, and striped mullet, a 3 order. And then using the non-transformed data, which again is more sensitive to rare species, we found the windowpane flounder had a 3-fold decrease, winter flounder a major 5-fold decrease, which I could have told you just based on the angling reports. White perch, 3-fold decrease, and then toadfish again doubled, striped anchovy, 3-fold increase, skilletfish, 2-fold. Skilletfish is an interesting fish. This is the one shown in the lower left, that cute little fish that looks like a tadpole got stepped on. And they were never seen in this area until a number of years ago. And all of a sudden they are clinging to pilings and rocks all over the New York City region. And they're a southern warm water fish. Striped burrfish have come on strong, and half-beak also. So we discern some major changes in these species, which is one of the outputs of this paper. Next slide, please. But the plots, or the output that really interested me, were the ones where we did multidimensional scaling. So if you look on the left panel, you'll see that across these years, I think it was 1989 to 2017, the years in terms of their abundance and occurrences are really tightly clustered. There's just a big swath of very clustered years that are not that different from each other, except for 96, which I don't have an explanation for. So we call that cluster A. Then cluster B, to the right of that, is far more wobbly. It's not tight and discrete. You can see there are a lot of changes going on, and they're focused mainly on the years in the 2000s and not the earlier years that were more tightly clustered on the left side of the image. So next slide, please. So we believe that's related to the Atlantic multi-decadal oscillation, or AMO. The AMO, you can see one of its phases in the map on the right, and you can see that the New York area is in some pretty dark red, and that's a firm kind of placement in an AMO of that period. Think of it almost like large scale, almost ocean-wide change like El Nino, but it's an Atlantic version, and it's smaller and different. So one of the ways we track AMOs, you can see in the lower left a figure that shows various phases of the AMO, and you can see that it wobbles back and forth. And right around the late 1990s, it went into a very strong positive phase, which means that it was very warm. And that warmth, we think, was expressed in the nature of the fish fauna. A lot of the species that showed changes were more typical species that live here year after year after year, like black sea bass or porgies or whatever. Next slide, please. If you look at the figure on the right, this is untransformed data. This is raw data. And this is the same data as in the left panel, but it's been treated differently. It just has not been transformed using this fancy sounding Hellinger transformation. So you see there that there's one major cluster around what's labeled as C. It's everything is pretty much within that same set of boundaries until we see a change in 2010 on the lower middle. And then in 2011, it starts going off to the side. In 2012, it is now in a different place. And 2012, you would think, well, okay, obviously that's Hurricane Sandy. That must have just mixed things up in some way. But it's not the case. In fact, in 2012, all of the fish sampling that was done by the state's survey was over already. So anything that was shown to have been occurring in that period would have been from post Sandy happenings. So in 2012, it went to a whole new location that we think was reflective of the famous marine heat wave of that period. And you can see that from 2012, it dove down in 2013, where you see E in the lower part of the diagram, to a unique location. And I think that unique location, in terms of all the other sites, was due to the repair that was occurring following Sandy in October of 2012. I think 2012, Sandy did a lot of damage or change to the ecosystem, and but it wasn't found in our data because our data was not coinciding with that. And then in 2013, you had the kind of response to Sandy off by itself. So the question then was in 2013, well, what's it going to go back to? Is it going to stay there, or is it going to go back to that constellation labeled as C, or will it go back to the D cluster? And in fact, it seems to have stayed back at the D cluster in a separate kind of cluster from what had been the long-term cluster. So we see a change going on, and we can track that change largely to the appearance of southern species. We are getting a regular trickle now of fish from the south that were not here before. Next slide, please. So this shows you the location of the marine heat wave of 2012, and you can see that New York was squarely in the middle of that. Next slide, please. An interesting aside, this is a bit of a tangent, but I've been fascinated by what happened with tilefish. Tilefish you've probably eaten, and it's delicious. They live in very deep water. And this photograph shows one uh, from 580 feet, which is kind of depth they're often caught at. Now, living at those extreme depths, they have very narrow thermal tolerance, and they live typically uh, around where the shelf meets the slope, all up and down the East Coast. And they were not even known to mankind until about 1880 or so. Then they were discovered, they were found that they could catch them in fishable numbers. And then suddenly there was this mass mortality where there were hundreds of, or maybe, or at least tens of thousands, if not hundreds of thousands of dead tilefish all over the surface over a broad swath of the ocean. And apparently there had been some kind of underwater event where cold water went up the slope and shocked these fish to death. And the point I'm trying to make from this is that there's a flexibility. Let me put it this way. These fish are common during times like this, but can be ravaged by nature unexpectedly. And that even though we think of the ocean as kind of a stable place that has ongoing dynamics that are real in terms of ocean movements and currents and all that, there can be these anomalies, and these anomalies now can involve very, very high temperatures that are changing our fish fauna before our eyes. Next slide, please. So some caveats in the Jamaica Bay survey, I think was really indicative of some things that are going on all around us. But it focused mainly on smaller individuals of large species or smaller species. It was not focused on, large individuals like 10-pound bluefish. You don't catch them in these nets. So, I think it was and is a terrific survey, but no survey is perfect. The gear is not ideal for every single species. And the survey is not conducted year round. That's not as fatal as it might appear, because in the winter, there's very little fish life in some of these shallow estuaries anyway. One thing that entices me about all this is that we have not looked at these same data for the last 10 years, or from the many, many other bays that were characterized the same way. So there's a rich database out there that is awaiting further analysis in terms of these questions. Next slide. So even though that survey focused a lot on littler fish, there are things we're seeing now with larger individuals. In the lower left, you see tarpon. Tarpon are now being seen in our waters every year. Not many, but it seems like they're there pretty much every year in low numbers. The redfish in the middle, channel bass, they had not been constant visitors, but there was one year about 3 years ago where about 2 dozen were caught in the surf. And I think that's the vanguard of what will come in the future. And lower right, this is an advertisement by a charter boat captain for epic cobia fishing on Long Island, which sounds like an oxymoron. If you tried this 30 years ago, people would have laughed. But cobia, which are these big fish related to dolphin fish, are now being targeted and caught in reasonable numbers around Long Island. So the changes are across different size classes. Next slide, please. But you can't figure out everything. There's some ongoing mysteries that intrigue me. One is the black drum on the left. This was a species that used to reach weights of 60 or 70 pounds around New York and was common in the 1800s. In fact, they were captured in the Harlem River, of all places. and they pretty much disappeared through most of the 1900s, but they're making a bit of a comeback now. They were numerous enough and large enough in the 1800s that they would eat the oyster shells of the oyster planters in Staten Island, and the oyster planters would actually hear at high tide their shells being crunched by these fish, which have the power to actually crunch large oyster shells. On the right, you see a striking fish, a sheep's head. This is a 17-pounder that was caught this year at Rockaway Inlet. And this is only 4 pounds smaller than the world record for the species. And sheep's head were very common in the 1800s. I found an account of farmers after the planting season going offshore on reefs and fishing for sheep's head commercially. There were that many of them. And they disappeared for most of the 1900s. And they're making a comeback now, particularly in Jamaica Bay. Next slide, please. One brief tangent on crabs. I had a student, Jessica Satrohan, who worked on the same DEC data on crabs for all the bays that we had data on, but just focusing on Jamaica Bay now, you can see that over this time period, there are a few interesting things. One of them is that the rock crab, which was and is a true cold water crab, has gone from very high numbers around 1990 to essentially nothing today. Growing up as an angler, I would catch them very frequently. Now I never see one anymore. I think they are a casualty of colder waters. Warmer waters. And below that, you see the spider crab. Spider crab is also a cold water crab, and it has the same pattern. Around 1990, its numbers crashed, and we don't see much of them anymore. Other interesting things are we have for the Asian shore crab, which is an invasive species that came in the 1980s, that it did a lot of wobbling back and forth, but like many invasive species, it becomes it's a little bit out of control in the beginning, and then it becomes less egregious over time. So its numbers have sort of hit a balance. And then for blue crabs and lady crabs on the top, those numbers don't really reflect Jamaica Bay per se. These are species that are living in the ocean waters and spawn in ocean waters, and then each bay gets some contribution from those spawnings. So I don't consider that reflective of Jamaica Bay. And let's move on from there. Next slide, please. So what's to come? Well, our data only extends to 2017, which is now quite a while ago. We've had a lot of warming since then. And I found a couple of quotes that I thought should sort of wake us up to this threat. This one says, from Greenland to the Sahara and across to the Americas, the waters of the North Atlantic Ocean warmed at an unprecedented rate in the summer of 2023, and that the intensity of the warming in that single summer was equivalent to about 2 decades worth of warming for the North Atlantic. So this is not slowing down. If anything, it seems to be accelerating. And I think the changes that I've documented today are just, the tip of the iceberg. There's a lot more change to come. Next slide, please. So what's to come? One way of looking at this might be, well, we're going to have Chesapeake Bay in New York real soon. Chesapeake Bay has a constellation of species like the croaker in the upper left and the redfish in the upper right and sea trout in the middle and tarpon I already mentioned and some others that are sliding northward. And I think we're going to see kind of a Chesapeake Bay fauna here eventually. I don't know how long that will take, but we're going to lose some of our beloved species like winter flounder, and we're going to pick up species that we're not used to seeing or catching. Next slide, please. So some final thoughts. I think it's best to somehow annually monitor fish communities, and I hope the DEC survey goes on forever. I think it's interesting and useful to place the results in a regime change framework and to look for wobbliness in these data to forecast tipping points, which may be relevant to monitoring or regulating both commercial and recreational fisheries. And one other point that I think is important too, that we should think more deeply about is species loss versus species replacement. We're going to see change, but change does not always equal harm. I'm not advocating warm water or major heating, but we're always going to have a fish community around us. And the question is, what exactly is going to look like? A case in point is I live fairly close to Hempstead Harbor in the Western Sound, and there's a there's a pier there. A lot of fishermen fish off, and for the last few years they've been catching lots of porgies, and they're totally happy with their porgy catches. Then last year and this year, their porgy catches were replaced completely by spot, which is a similar size species. They are perfectly happy catching spot and not catching porgies. So, I think there is an underlying question here as to what's interesting versus what really matters. Next slide, please. So I'm going to end right there with a bad joke. I hope you have some questions and comments.
Thank you so much, John. That was an excellent presentation.
Thank you.
Maybe I'll start with Alex Mercado. He asks, can you suggest why the numbers of silver hake, window pane, mackerel have continued to decline in Jamaica Bay when they are relatively abundant further east on Long Island? I think your presentation touched on that a little bit, but
Well, I would not say those particular species are that abundant anywhere in the New York area. I think they've all declined. There's not a lot of great data on the particular species, but I don't see a strong difference there. So maybe I don't agree with the premise. Do you have a theory on that?
Yeah, I guess I was asking because I work with Cornell Cooperative Extension out on Eastern Long Island, and we do
Right.
some inshore trawl surveys, and we do see those species like in fairly significant numbers. Maybe not what they have historically been, but I was kind of wondering if because of the context of this talk, if you know of like in the Western Long Island, if there's like a lower population dynamic, specifically with whiting, window pane, and mackerel. I mean, I see window pane a lot. Again, it might not be a historic amount compared to what it used to be, but we do see them probably every seasonal survey we do in fairly significant numbers.
Yeah, these species don't shut off, from abundant to nothing overnight. It's a process where the waters become less and less amenable to them. And, I think we're seeing some differential reductions over time, but with strong trends towards probably final extirpation from the region. And you have these strange comebacks, like mackerel came back out of the blue after 25 years of not seeing mackerel locally. They had one really good spawn. I saw it 2 years ago that there were some young ones around. They called them tinker mackerel, which I hadn't seen for decades. And then boom, sure enough, there was some major large fish the next year. But it's not sticking. It's one of these. It seemed like an ephemeral kind of response to really unknown factors. There's so many things going on besides the physical aspects here. We talk about the physical aspects of temperature, but we're not really bringing in major factors like competition and any kind of feeding trophic issues.
all right. Our next question is from Don Reapy. He writes, in the last few years, I've caught striped blenny in my killie trap. Before that, I never saw them before. Species change? What are your thoughts?
Yeah, I don't know. I haven't paid attention to striped lennies. I don't have my own sampling program. I think that it would be useful to look at the DEC's database on that because if they are around, they would be picked up. Another place that gets some unusual fish sometimes is the River Project at Pier 26 in Manhattan. They undergo an ongoing trapping, killifish trap survey, and they catch some interesting species just like that. So you might dig a little further, but I don't know offhand on that species.
Cool. All right. Emilia Lepi asks, regarding your last slide, when does change equal harm? What do you think is the tipping point, specifically thinking about conservation efforts?
Well, I'm not sure what you mean by the tipping point. You mean when change equals harm? I think that the fish communities will sort themselves out, and barring nuclear disaster or huge oil spills, we're going to have fish. It's just going to be a question of how are they allocated among the species, and that remains to be seen. So it's kind of hard to say where this will go, but I do want to just underscore how important anglers are in kind of sensing the environment and what's going on out there. You can mount a little survey, it costs a lot of money and you learn some things, but every day there are thousands and thousands of anglers fishing out there. And fortunately, we have some literary avenues for them to, share their results. So we learn a lot just from this army of anglers out there who are constantly sampling the environment, and I think that's a really great thing.
I actually wanted to jump on that real quick and ask you a question. Have you thought about pulling in data from iNaturalist to kind of supplement the data gaps that the DEC has?
Yeah, I love iNaturalist, and I use it in my teaching for vegetation and occasional weird birds, but I don't. I have not done that, and sometimes when someone catches something weird, the fish, they will then post it, and people will respond, and then you learn about it that way too. But I should look a little further at iNaturalist. I think it's a very useful program. If you haven't used it, it's a wonderful way to identify things you can't otherwise identify easily. But I was using it yesterday in my ecology class.
Yeah, it's a great program. All right. Isabel asks, are there any species that we're losing that you would consider keystone or critical to food webs?
That's an interesting question. Only from the point of view of human resources. In the Gulf of Maine, if we lose lobsters, it ain't going to be pretty. There's a lot of money being made in Maine on lobsters, and it's very clear that it's really important to the economy and to the tourism. But the center of abundance of lobsters is moving north each year as the waters warm, and eventually we're going to lose productive lobster fisheries. We had a really large fishery in Long Island Sound for lobsters that has largely disappeared. And there may still be one or two people out there trying, but this phenomenally lucrative fishery is now pretty much gone. So I think it comes down to the relationships of these organisms to people in response to that question. If you have water with oxygen and food, there'll be fish out there, and there'll be some of them will be happy, living in that world. But how does that affect humans? I think that's where we have a harder kind of problem. And it's important to remember too that we don't actually manage fish; we manage people to manage fish. We have all these laws that attempt to do a good thing in a very kind of cryptic and nebulous environment.
All right.
Anybody else?
And the next question we have is, any insights about the American eel in Jamaica Bay or elsewhere?
Yeah, eel have gone down a lot, remarkably so, I would say. And there are some attempts to get them further into headwaters that they need to live for the 20 or 30 years they live in freshwater or brackish waters. So there's an eel ladder on the Bronx River, but these are small scale programs. Overall, eels have declined dramatically. I had a master's student, Peter Malady, who looked at some changes in the fish fauna between the 1930s and the present day, and he found that, and this is around Long Island, he found that most individual trawl or seine hauls in the 30s caught at least 2 or 3 eels, and that when he did this several years ago, he caught 1 eel for the entire season using the same gear. So there's no doubt that eels are way down, and it's not entirely clear why that should be such a profound change. Most people don't eat eels anymore. They were popular when Europeans first came here, but most people don't touch them. There may be changes going on in oceanic conditions. It's just not apparent what the problem is with eels, but there are some encouraging signs that they're mounting a little bit of a comeback. Was there another question there?
Yeah, they had a follow-up actually. The same poster said the ladder broke a few years back. New York City parks are still using eel mops and translocating glass eels and elvers.
Okay, and they're translocating themselves too. Eels are incredibly, I hate to use the term creative, it sounds human, but they find ways to get over barriers. They can travel through wet grass. They will go through cracks in dams, and they can also, and I've seen evidence of this, adhere as little glass eels to the side of a dam and just used capillary action to stay attached to the dam and get over it. Also, with low dams, when you have high waters, they can sometimes get around the edges of the dams that way. So, in the Bronx River, which I've worked on a lot with some colleagues, we found that there were a large number of eels that were below the first dam that probably had a strong wish for getting above that dam. The second dam had a ladder for a while that now is not working. But meanwhile, we find eels through the third through the eighth dams that have no passage on them. They have just found ways to get up there. And what you find down low in that system is that there are large numbers of very small eels. When you get up to the first dams high up in the system, there are low numbers of very large eels. So they're fascinating species to study in that respect.
All right, another question we have from Nick. Early on, there was mention of a study showing 338 native species of fish locally. Does that include the visitors from warm southern waters? If
Yes,
so,
yeah. If it was caught once, it was on that list. Now that list is a little bit dated now. It's from 2002. Tom Lake, who um, keeps track of fish in the Hudson, has found species since that date that are not on that list. And it's a typical kind of vagrant situation. We're not getting mass invasions by anything. What we get are one or two of something new, and then maybe a trickle, and they come on kind of slowly. But Tom has documented 229, I think it is, species in the Hudson, and Briggs and Waldman had 338. There's a lot of overlap between those two, but we do have a very diverse ichthyofauna.
our next question is from Justine. The question is, I'm currently studying the Gulf of Maine through a climate change master's program, and 99% of accelerating warming in Maine is due in part from the reduction of the Labrador current. Are we seeing the same reduction in cold currents in our oceans in New York area as well, with an increase in warmer Gulf water?
I don't know about the current patterns. That's not my strength. What I do know is that the data is showing that waters are getting warmer, and they're getting warmer quickly. Uh, but as far as the details of the particular currents, I'm not sure. But I think that's incredibly important. There's a lot of concern about the Gulf Stream weakening, and that someday we may have a very weak or non-existent Gulf Stream along with a frigid Europe, because Europe depends on the warmth of the Gulf Stream. It's a great question to dive into, but I don't have a great answer for you.
We have time for one more, and there's something in the Q&A which might be a good way to end.
Yeah, it's a great question up in the Q&A. So an anonymous attendee asks, John, what is your favorite fish for Jamaica?
God, I'm going to have to break that up into freshwater and saltwater. I would say freshwater, I love landlocked salmon. If you hook one of those, it just jumps all over the river, and they're incredibly strong and beautiful to see. In marine waters, I've been a striped bass angler and addict for almost 60 years. So that's my marine fish. So I can't bring it down to one, but those are my top two. Can't go wrong with them, either one.
Mm. Excellent. So we're right at time. If I want to start by first of all thanking John for really just a fabulous presentation, and also like the Q&A was really insightful. So thank you very much, John, for that contribution.
Thank you.
I also want to thank Alex for co-hosting and for facilitating the webinar today. So thanks, Alex. Been a great partner. And I failed to mention at the top Thomas Morton, who's a master's student at Brooklyn College, who's been helping to organize these webinars. And I want to end by thanking all of you for attending, for asking great questions. So have a great day, everyone, and we'll see you next time.
Thank you.
Yeah.